All right, well, I guess it's time to get going since all of you guys are here. Welcome to today's presentation on how to clean contaminated TCs. It happens. It happens to all of us guys when you're working in the lab. Look, even when you're working with a flow hood in sterile conditions with aseptic technique, doing the best you darn can, things happen. As a matter of fact, I pulled out a jar of distilled water today that had two clumps of mold growing in it. They're straight from the store. Had two clumps of mold growing in it. So always autoclave your water. Even if it's straight from the bottle, don't watch the stuff on the internet that says open it up and use it. You're going to end up with a contamination. That's also the reason they tell you to change your media every 2 to 4 weeks, because everything takes 2 to 4 weeks to start showing signs of growth. So as long as you're changing it every 2 to 4 weeks, you never notice that something's growing. Anyhow, moving on along, I've brought two different cultures for you. Now, this one here, I just put in, let's see, it says on the 2nd of this month. And I'm going to show you as best I can on camera here that there is some funky stuff growing right at the base of where it's inoculated. Can you see that white fuzziness right here? Point is, there's a little bit of fungal contamination right here at the base, and if this happened to be a unicorn plant that you wanted to save, you sure don't want to just throw this thing out, especially if it was something that you purchased for a few $100s and you're trying to multiply it, which when you're doing TC work, guys, generally you're doing stuff on your unicorns. You're not doing it with your simple basic old house plants that you could get any old where. There's a reason that you're doing this, and it tends to be kind of expensive to just throw it out. So we're going to show you how to clean that up. Now this one, if you'll notice the discoloration in there, this is a bacterial contamination. It's probably something from my beard or a piece of skin flake or something that fell off or whatever it is off of your hand. I mean, but generally the bacterial stuff comes from you. You or something in the air. Maybe I didn't brush well enough that morning and breathed on it just wrong. Who knows? Maybe I didn't wear a glove. Maybe I sweat a little. You never know. It could have been any of those types of things. But bacterial contamination, generally, I would tell you throw it out, right? If you've got bacteria, you don't want that stuff spreading, throw it out. But there again, for the purposes of these videos, or of this video, it's to show you how to clean it, right? And how to reintroduce it. Also, if you'll look at this, the specimen's beautiful. It's got a brand new leaf popping out in it and everything. There's no reason to just throw it out other than the fact that there's a contaminant on the surface of this explant. All right, so we're going to go ahead and take care of that. It's a fairly straightforward process. Rock and rolling. We're going to start off with right here with this. This is a Euphorbia tismaloides variegated tricolor. So if you guys have seen any of our tricolor variegated devil's backbone, that's what we're working with here. And it just happens to be a contamination. It seems to be a fungal contamination that we want to take care of. Let me see if I can get that in the camera real good so it can see what's happening. Fungal contamination, number one. All right. Now, anytime you're doing that, you never want to shake this media. Let me come back to it. You never really want to shake this media too much. You don't want the nutrient solution and the contaminated solution actually getting all over that explant if you can help it. This part I generally do not do under the hood. I like doing this part of it outside, to tell you the truth, because of the fact that I don't want any of these spores landing in my laboratory. I'd much rather them be outside and then be able to clean up and come back into the lab here in just a little bit. But for the purposes of this video, we're going to do it right here in this room. So that's how that's going to work. All right, now the first thing I'm going to do is we're going to use isopropyl alcohol. This is a 70% isopropyl alcohol. You guys probably already have some of this sitting around your home, right? And what we're going to do is about a 30 to 60 second dip and swoosh before we do any cleaning on that. Let me grab my forceps out of the sterilizer. Got that. We set those down for just a moment to cool off. Actually, I'll tell you what I'm going to do. There you go. Now I'm cool. All right, we're going to open this up, pull this guy out, and we don't want it to touch anything else. So actually, you know what? It's got that contamination sitting right on it, so I'm going to set it on this tray. We're going to clean that up before I go to putting it in the alcohol. And the way we're going to do that, I've got a scalpel and the forceps themselves, and we're just going to get that right off. That's a chunk of the agar that's got the contaminant on it. We just want to get all of that off so it doesn't end up in our cleaning solution. Try not to touch anything else with it. And just get as much off as you possibly can. If it's hard, if you had some root material here, you would definitely want to use a brush. I generally use a little toothbrush for this, just to get it off of any of that surface and clean it off real well. Right here at the apex, where any of the branches kind of meet, that's where you'll see kind of some clusters, just like we've got right here. And yeah, we're just going to scrape that off. That's what we're going to do. I'm going to set that off to the side. Now, move that over a bit. We've got 70% isopropyl alcohol, and we're just going to drop that sucker in there for just a moment. Generally, I like to set this here for about 30 to 60 seconds. Give it a good swish around. Alcohol kills a whole lot of things, folks. Whole lot of things. It's also hazardous to your plant. Very hazardous to your plant. It'll eat that plant right up. I'm looking for jar number 1. There it is. Now, after you've got a good 30 to second swish, you will want to rinse it. Now, what I've done here is I've got 3 jars of sterile water, and that's all it is sterile water. And we're going to do a rinse. I have them labeled 1, 2, and 3 so that you can see which number of rinse. You don't want to dip it back into the same container, because then you're not getting the dilution of the rinse. All right, and it's not so important that we have completely sterile tools at this point, because you're not messing with anything else that's sterile either. Gonna set that over. I'm gonna put that lid on it and give it a good shake. Give it a good shake. This is wash number 1. Wash number 1, and all you're doing is you're diluting the alcohol that you just put all over it. If you don't do that part of it, that alcohol will dry up your plant, and it tends to eat it up just a little bit. Now we've got jar number 2. Same thing. That's all I'm doing, guys. I'm just going 1, 2, 3 here. I'm going to get my 3rd jar ready for me, doll.
Yep.
Pull it right out of there. Now the reason you're doing that is you're trying to dilute that. Come on, jar. Trying to dilute that alcohol just to make sure that it's not any residue sitting on there. But the alcohol is a killer. The alcohol definitely does the trick. Swirl it around in there. Can you open that one up for me?
Yes.
Thank you very much. That way I don't have to do that. Drop it in the 3rd wash. All right, I put the lid on each one of these to show them. Give it a little swish. Okay. Let it set for just a second. Now that's just water. All we're doing is getting that off. Now the second part of this is a 20% bleach. Okay, now we do a 10 to 15% surface sterilization with a 10% bleach, right? And we usually do that for about 15 to 30 minutes for the when we very first do the surface sterilization on the X-plant. However, that didn't do the kill. It did not do enough for us. So what we did is we bumped it up to a 20% bleach solution, and we're going to let it sit there for just a minute. Now, on a 20%, we're going to let it sit just until we get to the point of starting to see some white on the tips of the white on the tips of the plant, of the X plant. That's where the material starts to kind of die off, is what it is. Now, while that sits around, we're just going to give it a little shake in that bleach solution, and we're going to set it off to the side. While we do that, I'm going to come back to my isopropyl alcohol, and we're going to start the process over. We're going to start the process over with vial number 2. This is our Aglaonema Emerald King, and it's beautiful. I really would hate to lose that, right? I mean, it's just too pretty of a...
And it's pushing out something new, I see.
Definitely. Oh, yeah, it's pushing out a brand new leaf. Brand new leaf. Beautiful plant. So I'm going to take off the parafilm there. Always wrap your vials and your jars in parafilm. That really helps with any kind of funk that you've got floating around your house. Going to open the jar. So the first thing is isopropyl alcohol. Now I'm going to move that over because we're going to have to do a little bit of work on this tray. Always do. Pull off a little bit of the agar. There you are. And remove the X plant. Set it down. Tell you what I'm going to do. I'm going to dip that. This is our last one. I want to kill it before I expose any of that. Alright. I don't like spores in my air at all. Set that off to the side.
You want to take these away?
You can if you like. Thank you very much. Now, anytime you're working under the hood, we're not there yet, but always work from left to the right if you're right-handed. Don't cross your hands, and try not to cross anything over each other so that you're not contaminating any surfaces. Now, when we're outside of the hood, it's not that detrimental. And as you see, there's a lot of dead material here that's scraping right away with this. That means it's pretty damaged. This is some pretty damaged stuff, and it must be because of that contamination. It's like it just rotted it away. I'm going to say it's very similar to rot, kind of like to root rot, which you can take care of root rot as well with this same procedure a lot of times. If you have a plant that has root rot, there is some salvaging this. Now, for this particular explant, I tell you, all of this material has had some infection. I can see that it's really done some damage on that tissue. So what we're going to do, we're going to take and cut it right here.
Yeah.
We're going to cut that right off. We've still got plenty of tissue here to work with. We're just going to sterilize this instead of that ugly piece there, and just keep working from it. Scrape this stuff off. Any of this external stuff that you can get off before we go further. Okay, now we're going to put it in our isopropyl alcohol right here. Give it a little swish, and we're going to leave it there for about 30 seconds. Now, I think you can go ahead and take that tray. I'm not going to need that anymore. I'm going to go ahead and swish this around, leave it there for about 30 to 60 seconds. Pull out our jar number 1 for our wash. Now remember, these are just distilled water. Now that alcohol, you really want to get that in all the surfaces to make sure. So I'll put that lid back on, and I'll give it a shake. Now give it a shake. I like to make sure it gets in all of that surface. Here you go. You see, shake, shake, shake. Shake your body. All right, I'm going to bet that's about 30 to 60 seconds total. And we are going to pull that thing right out of there, if I can see it. Hang on now. There we go. And rinse it off. All right. Put the lid on that for me there, dear. Thank you. This is rinse number 1. Now, rinse number 1, this is my dirtiest rinse. This is the one I like to shake up. I really like to shake that one. I want to get all that alcohol off completely. I want to make sure that it's so diluted that it's not going to kill our plant, even though it should have done a big kill on whatever mold, bacteria, or funk that could have been in there. And now here we go to transfer to jar number 2, which I've set here. Number 2. Yeah. And for this one, we'll give it just a little swish. It's not that detrimental. It's more about just getting all the residual off. Okay. And on to jar number 3. There again. So it's 3 dips in water, just to get that alcohol off. Here you are. Okay. I'm going to give it a little swish and swirl. A swish and a swirl. All right, we should be good there. Now we're going to come back to our 20% bleach, which is your second sterilizer. You got alcohol, then we're going to go to a 20% bleach. I'm going to throw him in there with the other guy. They're not going to cross-contaminate each other, so we should be fine there. There you go. Now I'm going to put that lid on there, and I'm going to shake, shake, shake a little bit. And let it sit for a moment. Let's see how our Tidmaloides looks. Now there's some stuff floating around in that water. I don't know if you can see it, but there is some stuff floating in that water. That lets me know that even after 3 washes, or 3 rinses, and the soak in the alcohol, that we're still getting some funk off of there, that bleach is just kind of eating up. So we're going to give it a little swish, give it a little swirl, and let it sit. Like I said, this part, this is probably the longest part of the process, guys. It takes probably 15 to 30 minutes maximum before you start seeing white on the edges of your stems, and that's where the bleach is actually killing off that material. So we're just going to let it do its thing while we sit here and chat for a moment. Does anybody happen to have any questions at the moment? You can unmute if you like. We got a couple of minutes to sit here. No questions.
I'll ask a question. It's probably on everybody's. Yeah, I've got a past medical background, so I'm going to ask a question.
Sure.
You put you've got your jar labeled number 1 for each and 1, 2, 3 for your water. When you're done with these particular samples, you're going to start over with fresh.
Yes.
For the next time you go to use it, correct?
That's
okay.
exactly right. That's exactly right. Yeah, all we did right now was just a surface sterilization. We should have killed everything with that alcohol, and then we're just diluting that down. Now we're into the bleach. Now we're going to be doing another wash right after this bleach, but we do use fresh jars of water. Okay, fresh jars of water. Some people will use a bowl. I like to use jars because I can put my lid on it and shake them up. Now, one thing I will point out, and I probably should have changed that for the wash jars. Generally, I turn the little lid part here upside down because that keeps it from sealing when you pressure cook it or autoclave it. Okay, we don't necessarily want to seal our jars when we pressure cook them. We're just trying to keep them covered. So if you'll flip that inside out, basically, it keeps it from pulling that seal on there. Another reason you don't like a seal when you're going into a hood or a tissue culture, when you pop that seal, there's a rush of air that comes into it. We don't want that. We don't want that at all. Now, if just for these water bath washes and dilutions, I probably should have flipped that cap back over so that I don't drip all over the table like I'm doing right now. But for just alcohol and bleach solution, I'm pretty well okay with that for the moment. But yes, the answer to the question, yes, you always get fresh liquid, fresh water. I've got 3 more jars full of water sitting off to the side that are also labeled 1, 2, and 3. And I labeled them 1, 2, and 3 pretty much for the camera. It was for you guys. I don't normally do that for myself. I just wanted to make sure that you could see on film what everything is. So that's kind of the situation. I'm going to take a gander and see what we're working with. Now I do see that the aglaonema on the base is already starting to turn white, which means that fresh tissue, that's right here, that big stump right there, that fresh tissue is already starting to be killed off by the bleach. It looks like it's maybe the first one to come out. We may pull it out. Heck, we might just pull these at the same time. Let me flip over to the other camera and see if you can see a little bit better on this hand cam. Let me see, maybe not. But on the tithmaloides, here's the aglaonema. Here I can see that there's a lot of white growth. You can't see that any better. Okay. On the aglaonema, I can see that there's some white growth right here at the tip. So it's starting to die, or that material is starting to die off, that tissue. On the tithmaloides, I can see it on the cut node and at the edge. They're both starting to turn white. That means that we're getting good sterilization. The bleach is doing its job. 20% is a high concentration. I'll tell you now, 20% is higher than I usually use. 10 or 15 is what we generally do for the initial surface sterilization. You don't want to kill your plant with the bleach. You just want to clean it. If I have trouble like we had here, this is where I would go into cleaning it. I'll also say that on this particular explant, I can see some more of the material starting to die back. What I'm probably going to do is take another scraping on that to get any of that dead material off. Do a good thorough rinse again before we go further. So, as a matter of fact, I could probably do that right now with that aglaonema. Let the tithmaloides set for just a second. Can you grab me another tray or another steel tray?
Yeah.
Thank you very much. And she'll be right back with that for you. You know what I'm going to do? I'm going to put these in the sterilizer and grab another set of forceps. All right, got a clean set. There we are. I don't want to use that same contaminated set of forceps to pull these out of the jar. Okay. There we go. Like to cool that off, and here we are. I'm going to move over just a little bit so you can see a little bit better. Okay. And I think I've got another blade sitting around as well. We happen to have another one sitting around? Thank you very much. Yes, let me hold this up, see if you can see it a little bit better. Right here. If you'll notice, the edge of that tissue is completely white. It's been killed off. It's being sterilized. However, you can also see that right in here, We've got some brown discoloration. That's going to be some rotted material. I want to remove that before we go further. This Aglaonema it's pretty eat up. It was pretty bad. However, you can also see this new shoot looks beautiful. It looks beautiful. So if we can just get this clean, we're going to have a great little explant. It's just a matter of cleaning it up. It really is probably because I let it sit there an extra week. I was going to clean this for you last week when I had to, or what was it, on Tuesday, I think? Monday or Tuesday? And we didn't get to it. So here we are days and days and days later. And, you know, it kind of overtook the plant. And I'll go ahead and open that right up in front of you. There we are. Brand new set of, or brand new scalpel, just because I don't want to put the same dirty scalpel on it. I can always sterilize these guys later, but, all right. Now, the good thing is, this isn't coming off the same way it was before, which is good. That means that it's not as dead as I thought. It's just ugly. It's just ugly. But a thorough cleaning is what is required here. Just making sure you get as much of that dead material off as you can, especially around this little growth node here, because you're going to have shoots and roots coming from that. There again, this part does not have to be done under the hood. You're not at your final sterilization yet. Once you go to your final sterilization, then you're going to want to move to the hood before you take it out of that liquid. But at this point, you're still just cleaning. Still just cleaning. Imagine you being in the garden spraying some fungicide on it. That's kind of what we're doing here. We're just kind of cleaning on the outside. All right, now, because of this material being dead, I'm going to get back up to the camera real good so you can see it. We're just going to cut that right away. We're going to remove that outer section. And it should, yep, slices off fairly easy. And we have nice, healthy green tissue here. Nice, healthy green tissue. All right, we'll cut back just a little bit more. Do a little scraping just because I know that outer part is dead. There you get, notice we're never crossing hands, right? Everything stays on the left and everything stays on the right. Never cross hands. It's just good practice, guys, good practice. Even though it's not that detrimental at this stage where we're at, it's just good practice to do it. Now on this particular explant, what we're going to do here is I'm going to remove, see, I think I've got another set of, yeah, I've got another set right here. I'm going to remove this outer skin. Look, we've got funk growing all inside there. We don't want that. We're going to get rid of that. We are going to get rid of that. There we are. Any of this excess material that you don't need, that's where all your stuff hides. Hides in these little folds of skin.
Funky folds.
Yep, the funky folds. Funky folds. And sometimes it's hard to grab too. There you go. Just get that off. Get that off. About time to get that tismaloides out of there. Just a minute. All right. Well, we got that skin back, and it's a whole lot cleaner than it was. Now, before I go dunking this into the next segment, I'm going to put this back into the bleach for just a second, just to get any of that funky material off. It makes it a little easier. I could do a water bath, but I know there's still some funk there, so I want to go ahead and give it a little swish. Give it a little swish. Okay. And that's really all that needed. It's been pretty well taken care of. I'm going to set that here. And I'm going to pull the tismaloides out. Alright. Now on this tismaloides, did I lose a leaf? No. It's just sideways. That's a pretty little explant. Pretty little explant. Boom. We're going to chop the edge right there off, because it is damaged and dead. Now I don't see anything growing along this one, so we're going to leave that there. I am going to remove that lower leaf. We don't need it. We don't need that leaf. As a matter of fact, I'm going to get rid of these top two, because there again, we don't need them at all. It'll grow new, and that's one less point of contact for anything to be, uh, for anything to catch a contamination in it. Set that here. Okay. Now, what we're going to do next is a water bath. So we got number one. Yeah, number one. We've got number one sitting here. And this is still a little funky plate at this moment, guys. We're going to do them both at the same time so that we don't have to go back and do this again. All right. Set that one off.
Okay.
I'm going to remove this tray because I no longer need that. Let me sterilize these forceps real quick. Alright, which to sterilize, guys, I just put them in our bleach for just a second, and then I'm going to go stick them in I've got a bead sterilizer. Let me, well, I'll show it to you in just a moment when you move over to the hood. And that's generally what I use. I get you to remove this for me. Take that back in there.
Yep.
Thank you very much. Alright, now, this is water bath number one. Give it a shake. All we're doing here, guys, is getting rid of the excess bleach solution that's on here. Now, really, the amount that we used isn't all that that high of a concentration, especially now that it's diluted. But we still want to go through the 3 separate baths, and that helps us get any of that crud that we were sitting there scraping around off too. Sure does. I mean, it's just a rinse process is what it is. Oh, we're just going to go from jar to jar is all we're going to do. This is our new water. Now, in your bleach, I do find that it helps just a little bit if you will use a couple of drops of detergent, liquid detergent, and that's where the bubbles come from in here. A couple of drops of liquid detergent as a surfactant. You can also use a product called Tween 20, which does the same thing. Set that up. Oh, that's wrong jar lid. Hang on. Which I guess it doesn't matter, it's water. Give it a little swirl. Get that off, dilute that bleach. Dilute that bleach. You're going to have to get a new drape here in just a little bit. This one's getting wet. And we're going to jar number 3. This is our 3rd bath. Sure looks a whole lot better.
Mm-hmm.
A whole lot better. That Tidmaloides, I think, might just make it. Sure might. Now, the Agli Anema is pretty rough, but we'll see. We'll see. And there again, we're just shaking it up, giving it a bath, guys. Getting all that bleach out of the cracks, crevices, and crannies. Right, no nooks and crannies, hiding any bleach in here. Okay. Now, here we go. This is the final step of the process, and I like to leave this one for about 5 minutes. We're going to do them both at the same time. This is a product, let me see if I've got the bottle sitting around. I do. This is a product called Fizan 20. Yeah, Fizan 20. There's another product called Captan that works pretty well. They're a fungicide, biocide, and viricide. That it's a highly concentrated look. Even the instructions tell you, after using this, change your clothes, take a shower, change everything. Dangerous. Look, danger, danger, danger. It says it right there. So anyhow, the mixture I use for this type of sterilization is 8 milliliters per gallon, right? 8 milliliters per gallon. 5 milliliters is right at a teaspoon. It tells you 1.5 to 2 teaspoons, depending on what you're doing. And for plants that are a little bit more delicate like these, I mean, I've got brand new shoots coming out, and the others still had leaves on the Tidmaloides. I wanted to go with about 1.5 teaspoons. We've already done isopropyl alcohol. We've done a bleach sterilization, and now you're dipping in Fizan 20. Now, the Fizan 20 is much more mild than the bleach and the alcohol as far as killing your plant. This is made, it's a phyto, what do you call it, a phytosanitary product. I mean, it is made for plants. It's made for, you know, algae and bird baths and fountains and things like that. So it doesn't kill off everything except what it's supposed to kill off. That's why it's the 3rd wash. I want to kill everything with the isopropyl alcohol. I want to do a further sterilization with the bleach. And then the final is just to make sure that there's no fungicides, bacteria, molds, or viruses. And we're going to do it right here with some Fizan 20. Let me go ahead and put that in there. And we generally are going to let that sit for right around 5 minutes. And no more. No more than 5 minutes on the Fizan to soak. just to soak. Now this is the final wash that you're going to do. So that's number 3. Number 3. 3. Yeah, 3. Final wash. And give it a little shake. Give it a little shake, and you can remove those if you like. The next angle will be under the hood. So I've got the thing turned down now. There's no sound on that camera, so it shouldn't be too loud with the fan blowing. But really, if you wanted to do that in just a still air box, that would be quite okay. And the reason I say that is when we're dealing with a known contaminant, you could possibly be blowing those spores and bacterial pieces and particles around inside that your flow hood area. Just because you've got a flow hood doesn't mean that there's nothing growing in there. All it means is that you've got a rush of air moving past the object, right? The idea is that it pulls air. Well, mine is it's a top-loaded flow hood, but some of them are from the back, some of them from the top. But they'll pull air from one direction, and then it just pushes a rush of air past it so that there's no pathogens that are coming from any external source. Theoretically, the air moving across your work surface has been cleaned through that filter, right? But if it's just blowing around spores that you know are alive, you're just contaminating your work surface. So we don't do that. You could either turn it off completely and work in it, or work in a glove box, and you're going to be a lot more sanitary than just having that rush of air when you know there's a known bacteria or a known contaminant in it. That's just what I have found anyway, because then you don't have to balm your area later to kill it with some biocide. We'll let that sit for just a moment, guys. I think we said top 5 minutes, and it's probably already been 2 to 2.5 minutes. And then we're going to move under the hood and reintroduce them to culture. So we're almost there. Almost there. Are there any questions at this point, or has it been pretty straightforward? I mean, I try to make this pretty straightforward. Feel free to unmute and ask a question. Or not. That's okay. That's all right. Okay, well, we're about there. I think we've been in there about 3 or 4 minutes now, so we're going to go ahead and move it into the hood situation. All right, now I'm using my mobile setup, and anytime we do our classes or our workshops, I don't feel that a hood is good enough when you're dealing with 10 to 15 to 20 people working. So generally what I'll do is take a still air box and put a small mobile laminar flow hood inside of that, so that I only have to clean that area, and it works a whole lot better. I've noticed the results of the classes have gotten better using both, right, instead of just one. If you're doing it by yourself and you're in a nice clean room, you're probably okay with just the hood. But when you're dealing with multiple people and multiple things going on at the same time, and I don't know what conditions any of these people live in, I like to know that 2 feet around it is completely clean all the time. So then the only thing I have to worry about is what funkiness comes off of their hands. And there's not that much stuff going on with it, because we're going to put on some nitrile gloves, right? Now, before I put on the gloves, I like to use 91% alcohol on myself, just to make sure that my own skin is, as clean as I can possibly get it. Yes, it dries you out just a little bit, but I also know that there's nothing growing on my hands right before I go into the hood. Like I said, those are the things that'll give you a bacterial contamination, is stuff falling off of you. And if the only thing that goes inside of it is from here up, I want to make sure that there's nothing that's going to fall off. So we're good. We've got that on there, and I just let it dry, because that's how things actually sterilize. I'm using a 91% alcohol on this, but generally for a good surface sterilization, 70% works great. If I was spraying the inside of the hood compartment, you'd want to use 70% and let it set for about 10 minutes to make sure that you get a proper sterilization. Now we are about to go in. All right. And we've already got our tools inside there. I've got everything sterilized and ready to go. So, all right, let me move on over and we'll get there, folks. We'll get there. Moving to the flow head cam. All right, can everybody see that okay? We should be able to. All right. Now this is a pretty simple process. I'm just going to remove the lid there. Now our containers are open. I've got a few test tubes that are ready with our media in there. Now this media is not quite as thick as some because I want to insert it inside of it instead of sitting it right on top of it. So that's exactly what we're going to do. All right. Good sterilization on those tongs. Now with one hand, this is the idea, with one hand, guys, you're going to get good at this. Remove the cap, okay, and pull it to the back. So it's not reaching over anything when you do this. We'll reach in there and grab our X-plant as best I can see. Oh, come here, little guy. I lost you. There you are. Let out just like so. And right into the media. One hand, one touch. You saw that? One hand, one touch. And set it off to the side. We're going to do the same thing again. And you're never going to reach over any of your work surfaces with anything that could contaminate you. All right. Give it a little rinse. Now you do not need to rinse off the Fizan 20. The Fizan 20 is going to keep doing its job. It is made to be sprayed on a plant, a live plant. So you're not going to kill it. You're not going to do anything else. And here we are. We've submerged it back into the media. And voila, folks. That is the process. We now have two X-plants that we have re-sterilized and reintroduced into tissue culture media. Boom. That's the long and short of it, folks, right there. It's not a super tough project. It's just a little bit time consuming. If you want to do it properly, you're going to need to spend that 10 minutes, that 5 minutes, that 15 minutes that it takes for each stage to make sure that you properly get there. But that is the long and short of it right there. Hey, thanks for watching today's video. If you like this type of content, be sure and hit that like button. Be sure and hit that subscribe button. Share it with your friends. It really does a lot for the channel. It helps us grow and helps us show that this is the type of content we need to be creating. Other than that, thanks for being here, and we'll catch you on the next video.